um, for this audience. It's going to be about 2/3 into human speech and 1/3 into a new animal model that we're considering to understand certain aspects of interactive communication and human speech. So I'll start with the movie, and this is really your wheelhouse, not mine, but it's an example of a person with expressive aphasia that has had a stroke and was interviewed by Jonathan Howard, who's a neurologist here at NYU. Jonathan has made these movies available for educational purposes, and I'll show this movie to you.
And what happened?
Yeah.
You're not sure?
Yeah.
Did you have a stroke?
Yeah.
Okay. And what symptoms did you have? What's wrong with you now? Okay, so you can't move the right side of your body. Are you able to speak okay?
Well, yeah.
Okay. Do you feel that your speaking is normal?
No.
What did you used to do for a living?
Yeah.
Can you talk about it? You're pointing to the computer. I guess you work with computers, is that right?
Yeah.
Just talk about it a little bit if you can.
No.
You can't?
Yeah.
Does it bother you having trouble speaking?
Yeah.
So I think many of you in the room or in virtual space have seen many of these kinds of interviews or conducted many of these interviews. It's still quite heartbreaking to me to watch. This also affected my father-in-law, who had stroke and had very similar kind of language deficits. And so I want to, of course, follow this up by just making the simple point that, of course, human speech is a very difficult thing to achieve. You know, the fact that we can do it at all is still kind of amazing. Words have acoustic structures that require us to move our articulators at a 20 millisecond time scale in some cases. And this, of course, is the coordinated work of about 100 different muscles: respiration, articulation, etc. And we're not often speaking into the void, although when I'm here talking to my computer, I often feel that I am speaking into the void. Typically, when we talk, it's as part of an interactive act. And so one way of sketching out that interactive act is to, for example, as we did here with this 2-minute annotation of a podcast, there is a kind of light gray and dark gray person talking, and each symbol here represents the duration of that speech passage. You can see that they're interacting with each other. And then what is, I think, a real hallmark of these kinds of interactions is turn taking. And so this is an example of very rapid turn taking back and forth in these two individuals that are chatting as part of this kind of podcast interview. This has been examined really thoroughly by a number of groups, and this is, I think, from one of my favorite papers from Stephen Levinson's group at Max Planck in the Netherlands. And what Stephen and his colleagues found is that the turn-taking floor transfer offsets, the time between speakers, can be blazingly fast. And so just to kind of get you acquainted with this graph, the negative numbers here are times when one speaker is actually speaking over the other speaker, and these long positive numbers are these kind of long pregnant silences between speakers, which really, try it yourself with a friend sometime and just wait a full second in between turns, and it can seem like eternity. And so we're excellent at this. How does our brain actually do this is, something that I think is a really fascinating question. And so as being a relative newbie to the field of speech processing, we come at it from the songbird, so from a kind of neuroethological place. I started looking around for models of speech, what are functional models? And there was an extremely influential book, at least for me, and I think for many other people in this field, written by Frank Gunther. So this is Neural Control of Speech, MIT Press, came out a few years ago. But even in that book, this is something that really was looking at a single speaker generating a series of utterances and thinking in a kind of engineering way about feedback loops and forward models of corollary discharge and all of this. And I think it has really advanced our thinking. But in terms of models that exist for interactive communication, there really weren't very many at all, and certainly no prominent ones. So we tried to propose one. We worked with Frank, and the idea here is to first think about what's going on in this person's head during this kind of interaction. And so, of course, she has to parse the sensory stimulus, which is another speaker. She then answers this with the motor response, but speech is not a reflex arc, right? There's some kind of cognitive processing that's going on. And I should mention here, of course, that we do know quite a lot about the two bookends of sensory and motor, and I think a number of labs have done, I think, extraordinarily important work along these lines, Eddie Chang and many others. But what I was interested in are what are the kind of cognitive pathways that exist between that sensory piece and motor response. And so we had a paper that came out a few months back in the Annual Review of Neuroscience, where we proposed some kind of cognitive modules. And here we're basing this not only on behavioral work, so primarily out of the field of linguistics, scouring that literature, but also neuropsychology. So what are different aphasias or different types of brain insults that can selectively affect one function and not the other? And this can give us some idea that there can be separable modules that exist within the brain. So the first thing we think of is, if I'm responding, I have to have a message, this kind of content that exists here. I have to deploy that message with specific timing. So we talked about turn taking and being able to, with sub-millisecond precision, answer my conversational partner. And then also something about affect, right? So there's a kind of coloring to this message that comes from an emotional pathway. And of course, there can be affect-only vocalizations, things like crying, and we could talk about that if you're interested. But the idea here is, and I'll kind of first take a half step back and say, you know, all models are wrong, some are useful. We fully anticipate this to be the first step of many to try and get the ball rolling in terms of functional models. But I think this is also a way of forming a framework for what needs to be done to really understand how interactive communication works, and maybe even how other creatures can take different boxes within this more complex framework. And I'll talk about that in the second part of the talk. But for now, I'm very interested in these kind of more cognitive processes that enable the content pathway, and specifically with respect to response planning and interactive communication. And to do that, we worked with the University of Iowa, and so this is a group. These were the direct collaborators that we worked with there. Although Rusbe, of course, is someone that I got to know through this and is a fantastic scientist, and it's a real pleasure to speak to his community there too. And he's just a tremendous guy and a brilliant guy. You guys are very, very lucky to have him there. So what we wanted to do is look at the planning processes that are required for interactive vocal communication. And so the data that we're collecting to address that question is electrocorticography data. So this necessarily means electrodes that are directly on the cortex. And those electrodes give very, very clear responses, both in space and in time. But of course, you have to be broken enough that there's an opportunity to take part of the skull off and introduce these electrodes to the brain. So this is something that I want to say, first of all, this couldn't have happened without a number of patient volunteers, in this case, 8 people that were either undergoing treatment for medical intractable epilepsy or for the removal of tumors. And importantly, what I'm going to show you was constant across both of those two conditions. So we analyzed these separately, the tumor and the epilepsy cases, and we find that what I'm going to show you in terms of speech planning is true across both. Okay. What is amazing is in the case where there are recordings here, either in an acute environment where the patient is there and is recording right on the operating room table, or an extended acute environment where that person is being screened for seizures and is in a place for a week or more to monitor brain activity, they're still capable of turn taking. And so this was an example of that. And so we aligned the experimenter's words here. We look at the offset of what the experimenter is saying and saw the amount of time. This is just during a free conversation between the experimenter and the patient. We looked at the amount of time it took for that participant to respond. And you can see this is happening with very short latencies. And so that's something that seems to match, at least qualitatively, what goes on under normal conversational exchanges. So how do we look at planning? Because we think planning is really important for this fast back and forth, and people like Levinson and many others have shown that planning is important. You kind of have something cooking in your own mind before you can deploy that as a very rapid response. And so how can we actually operationalize planning? How can we isolate planning dynamics? And so we went to, I think, a really brilliant task, and one that was developed by a different group. Sarah Bogels was the one who came up with this task. We translated it from the original Dutch and expanded upon it. And we have 3 different types of questions that all follow the same kind of rule, and that's this. There is a series of words, and one critical word shown here in blue is absolutely key to telegraphing that answer or enabling that person to answer the question, thus initiating response planning. So if I ask the question, how many fingers does a healthy person have? As soon as I hear fingers, I can get my answer ready. How many toes does that person have? How many arms? How many legs? That critical word allows me to answer the question. There's also antonyms. So these critical words are the opposite words: soft, nice, sad, hot, and animal sounds: moo, oink, etc. Okay. So I'm going to show you now a kind of progression of what kinds of activity we can record using this task. Okay. So there's, in this case, an array, and this array is placed on this frontal and parietal region of our participant's brain. There's Broca's area here, so this is inferior frontal gyrus, no frontal gyrus up here. There's a speech sensory motor cortex, and each dot here represents one of the electrodes on an electrocorticography array. And the response is shown here with this color. So as the response gets bigger and redder, that means that in z-score space, that area shows more activity, and the activity is, in this case, defined as high gamma activity, which represents or is correlated with underlying multi-unit activity. And we filter between 70 and 150 hertz. Okay. There's also a strip here in this auditory processing region. So we have this large array and then a strip in auditory. The question, so what I'm going to show you first is across all of the questions that we administered for these, for this specific participant. This is the average response 200 milliseconds before the onset of the question. So keep your eye on the shaded region up here. And you can see these electrodes are kind of cold. They're blue. They're, if anything, dipped slightly below 0, and that's because our baseline is the entire session. Okay. Then the question starts, and we take the first 200 milliseconds of that question, and you can see there's activity here in auditory cortex. These electrodes light up. They're bigger and redder. Now we look at the activity. Again, you can see this sliding gray shaded region goes over to this critical information. And as soon as that critical word is given, we see activity emerges ferociously over in this part of the brain, which then seems to be 200 milliseconds before the response, transferring over to the sensory motor cortex. This is 200 milliseconds of the response, and this is following the response. Things go very rapidly back down to a baseline time. Okay, so let's go through that movie one more time. So this is a kind of baseline before I hear the question. Something is bubbling up here in auditory cortex. The critical word happens 200 milliseconds before the onset of a response. The response itself, done. Okay. So we wanted to try and characterize these electrode responses, and we thought that the three kind of clear categories could be perception, planning, and production. This is what we found to be the case when we were looking at the data in a way that I just took you through like this. So our idea was that if an electrode were a perception electrode, we would see plenty of activity during the question itself, because this is a sensory stimulus and we should see some kind of a sensory response. We created a model that could pull that out and say, give us those electrodes whose responses seem to be high during the question itself. And we see electrodes that look like this. So this is a single trial, and this is high gamma activity. And we see that it's active during the question. Okay. We see other electrodes that seem to be triggered with this critical word. And a trick that we played here was to actually ask the question in two different ways. One is to have the critical word in the middle. The early variant of the question would be the opposite of soft is what familiar word? Or to ask the question with the critical word at its end. The opposite of what familiar word is soft. And we see electrodes that seem to be following that critical word itself. And thus, we would call those planning electrodes. And we also see some electrodes that are production electrodes that are active during that response. The person here says two eyebrows, and that follows like this. Okay. So these are those same exact electrodes, but now instead of showing you one trial, I'm going to take you through all of the trials we performed for that given participant. So this was the electrode that showed perception responses. And so the beginning of the question is shown here. Every line is a single trial. The response itself is shown by the color. So these kind of more yellowy colors mean that there's a large response in z-score space. And the beginning and the end of the question are shown here. We've sorted by the question itself. And if we look in this electrode that we think looks like a perception electrode, every single question seems to light up that electrode like this. Now, if we look at an electrode that our algorithm is calling a planning electrode, we see something different, right? It's not on during the entire question. This is the onset of the question here, this white bar, right? There seems to be activity that's sprinkled across that. And then a production electrode, again, doesn't appear to be on here. The person isn't vocalizing during that question, so we don't expect to see something. If we organize the data in a different way, and now organize it by the onset of that critical information that we think could be important for isolating planning activity, now our perception electrode doesn't really follow this. This is the onset of the critical information in blue and the response in red, and we see that things are not well described in this perception electrode, but this planning electrode seems to be very well described. So the onset of that critical information to the response, which are this second set of ticks, we see a lot of activity at the level of this electrode. The response itself seems to be here in this production electrode, and then that again makes sense. The response equals production. Great. So where do we see these different types of responses? We have certain prior information, certain expectations. One expectation of sensory is that we'd see it here on the temporal lobe, as well as an island that's also been described to exist here in the more frontal region. Indeed, this is exactly what we do see. We expect to see the articulatory responses in the sensory motor region. This is where most of them are isolated to. For planning, it's anyone's guess. We're the first to do electrocorticography during this kind of interactive speech task. And so the kind of planning, we were guided by a number of different papers to look in a number of different areas, and we find there is this hotspot that appears to be happening in this more frontal region. We could put a finer point on that frontal region by just simply doing spatial analysis of the percentage of active electrodes in each neighborhood of the brain. And we see this very clear hub of activity, this kind of interactive planning hub, if you like, that includes the caudal middle frontal gyrus, this caudal portion of the inferior frontal gyrus, auricularis and triangularis, and then this portion of the ventral, in this case, precentral gyrus, a little bit of the temporal lobe. Okay. So our next question was, are these planning signals that we're seeing generic for any kind of planning, or is it language specific? So we can see that this area lights up for planning linguistic interactions or planning linguistic behaviors, but to what extent is that generic for any kind of movement? So we decided to do a series of experiments where we start with a generic premotor plan that doesn't involve any muscles involved in articulation. Of course, it would be involved if one was doing American Sign Language, and I think that's a totally fascinating avenue of research. But for now, for these 8 patients, we wanted to ask, with your hand, click a button 2 times. Okay, so they're planning and executing a button task, and we don't see that area light up in that case. Okay, so let's now engage articulatory musculature and ask if this area is engaged, but non-speech. So we can do sticking out your tongue, pucker your lips, smile for the camera. And if we do that, we don't see this area engaged. We don't see the same level of activity of those electrodes. What about any kind of speech? We start to see certain aspects of this light up. And now finally, what about with more overtly linguistic tasks that require lexical access? I'm going to hand you something. You have to go and retrieve a new word, do some kind of computation on the word. So here, the example is pluralization. Say the plural of goose clearly into the microphone. The answer is geese. And when that is said, this whole area lights up. So our kind of idea here is that the planning network we've identified is preferentially active during linguistic tasks. So the next thing we wanted to do, because maybe people aren't sitting in rooms and asking each other what the opposite of soft is, maybe certain rooms, but for me, we wanted to find out if the same kinds of findings that we had in the study could be extrapolated out to unstructured vocal interactions and conversation. We're working now with an expert in this space, Jelena Krivokapic, and she's been fantastic to talk about this and to think about how this can be expanded into conversation. We took a few steps forward in our paper looking at this, and I'm not going to go through all of the analysis, but I'm going to give, I think, a very compelling example. And if anyone else wants to read some of the other steps that we took, there is in our paper earlier this year, we put a fine point on conversational exchanges, and at least the kind of upshot of our analysis there is that things really do appear to be conserved from these structured tasks all the way to natural conversation. But this example, I think, is really cool. It's just raw traces, if you like, high gamma traces here rectified, shown from a blue and a red electrode. This blue electrode is here in inferior frontal gyrus. The red electrode is here in this motor production region. And let's first look at the red electrode. So here we're just chatting with the participant. Were you born in Mexico? She says, New Mexico. Have you been to Mexico? No. Never? Never. The red electrode is active during her speech. That's fantastic. That makes sense. Motor does motor. But what I'm absolutely fascinated by is this blue electrode that we think has a role in planning. And we see that that's active during our speech, during the experimenter speech. Were you born in Mexico? And you see this kind of planning signal tick up as the question is being asked. Have you been to Mexico? Again, a planning bump. No, never, never. Okay. So now we wanted to ask the question, are the planning signals necessary for normal vocal interactions? And again, I'm speaking to a crowd of experts. I'm still learning, and I'd love to hear from you, your own thoughts. But of course, one could look at this with retrospective lesion studies. We know that Broca's aphasia is a global language production deficit. So this is the first brain of Broca, with Nina Dronkers doing the anatomical analysis a few years ago. And our planning sites are shown here, and we've done our best to kind of circle a reasonable similar area across these two. We wanted to find out if these effects could be specific for speech planning. So one way we could do that is actually administer direct cortical stimulation right on the surface of the brain. And this is something that is reasonably common. So of course, Penfield did this in the 30s and gave us the whole concept of a motor homunculus. And this is some work that I think is just very consistent with what they found. We can stimulate this area here right at the juncture of the motor and somatosensory cortex, and ask our interactive questions. So these are our critical information questions. Which frequent word is the opposite of bad? Good.
What frequent word is the opposite of bad?
Good.
So what we're going to do now is administer stimulation, and that stimulation is given with this yellow line. So it's constant stimulation during this yellow line administered to this area shown with the red star.
The opposite of bad is which common word?
So you can see there's dysarthria here. The person is unable to get this word out. Their tongue feels frozen and unable to really answer the question in a clear and coherent way. Now, if we stimulate some of our planning areas, we'd expect a few different things, right? First of all, we wouldn't expect to see articulation problems the way that we saw the stimulation of this motor strip. But maybe we'd see some planning issues, right? We might see delayed responses, reaction times. We'd see speech errors because of an inability to do lexical retrieval, to try and grab that correct word. And indeed, that's exactly what we do find. So this is an example of stimulating in the same person. In fact, it's exactly the same question. The opposite of bad is what common word? We're now stimulating down here in the inferior frontal gyrus.
The opposite of bad is which common word?
Easy. Good.
The opposite of hot is what common word?
2.
So you can see this is kind of matching the description, some of our predictions. They can still speak during the stimulation, but now they're starting to get some questions wrong. They're getting the wrong answer. And in fact, what's I think pretty fascinating about this second case is the answer to the last question was 2, and they're holding on to that answer, and they deploy that answer here with this question. Here are 4 other examples, 2 each from 2 other participants, of stimulation-induced errors in responses. Which word is the opposite of fast? Rapid. That's during the stimulation. Stimulation stops. The opposite would be slow then. Clearly into the microphone, see the plural of foot. This is during the stimulation. They return foot. Okay, they were not changing the word or performing any computation on that word. They're just handing the word back. It becomes a repetition task. Now stimulation stops. What's the plural of foot? Beat. They get it right. This is our one person where we've seen a change on the right-hand side. This is up in MFG. The opposite of hot is what common word? Cold. Okay. Stim starts. How many toes does a woman have? 10. What animals who bark are often seen on farms? My brain just farted. So in addition to these kind of errors, we also see profound changes in the response timing. So this is control for a specific participant. You can see that their responses are quite fast. So on average, really right where normal responses sit, that 200 millisecond level. This is 146 milliseconds. With direct cortical stimulation, this balloons out to over 3 seconds during that state. And we can create a map. The real estate that we're most interested in is actually the area that we've done the least amount of coverage, but such is the nature of the beast when it comes to human subjects. We kind of catch as catch can. We're very grateful for any data that we get from these individuals. And so we're kind of still filling this in with more data. But right now, we've got 28 sites from 16 participants. And we found motor deficits at these 3 sites, which are all located here within this kind of motor or somatosensory cortex. We see these slower reaction times here. And what I think is also tremendously fascinating is that we see even faster reaction times outside of this area. Why that works, how this works, is something that I think is great. And it's one of those experiments where we can answer one question and 10 more questions pop up. And I think it's a fascinating direction perhaps for the future. So in this part of the talk, and again, I'm going to have a kind of coda that comes along where we take our first steps towards looking at this in non-human animals. Speech perception, planning, and production are separately represented in the human neocortex. A language planning network necessary for interactive speech includes the middle frontal gyrus and Broca's region. This network is tuned to linguistic operations rather than finger pressing or orofacial movements. Consistent cortical responses occur during scripted tasks as well as this natural conversation, and we can perturb this with stimulation, and that leads to slowed interactions and errors. Okay, so returning to this framework for how interactive vocalizations can work, one advantage of this is that instead of saying, hey, it's just a black box in there, we've created these other kinds of boxes that at least make the problem somewhat multifaceted and allow us to maybe hit this piece by piece. And as a systems neuroscientist, I think it is a little frustrating to have a box and arrow plot because we really want to get in and think about the neural circuit algorithms that are relevant here, right? And get to a kind of mechanistic answer. But language is unique to humans. It's really hard to throw your arms around something that has 86 billion brain cells. So we have solved this problem by examining a lot of different creatures in the lab that can do amazing vocal things. And I think what is great is those amazing vocal things aren't all of linguistic processing, but could maybe look at specific boxes here, right? And so this is an example of a songbird, and songbirds really can't solve the content problem, but they can deploy their song with very specific timing. I think what's going on here could be affect-like in reasons that I'm happy to talk about. And there's sensory and motor processing, clear song imitation and learning. This is a sonogram of that bird. And we've done some work over the last 2 decades or so to kind of take this apart and really understand how the bird does what it does. And this is an example here in the upper right of electron microscopy, looking at the wires, understanding the parts list for how the bird does what it does, and can really think about this at 9 orders of magnitude of length scales. So this is nanometers. Conversation in humans is really meters, right? And so trying to really dig down and say, how can a group of cells enable a song that can be learned just like babies can imitate their parents? And how can that be involved in kind of back and forth turn taking? And in terms of turn taking, this is a new animal that we've considered. That's a singing mouse, or Scotinemus tiguina. That's a funny name, and that is a funny animal. This is native to the cloud forests of Costa Rica. It's not your standard laboratory mouse, nor is it a different strain of mouse. It's a totally unique and interesting creature that was kind of ripped from the pages of neuroethology and now studied in a neuroscience lab to really try to unravel some of the wiring and some of the basic principles that enable the singing mouse to do what it does. We have a whole team. This was started by Steve Phelps, who's a fantastic collaborator at the University of Texas, a couple of computational guys out west, and then a team of researchers that are collecting data based on this fantastic animal. This is why the animal's fantastic. So this guy will start to do his advertisement call or his song. This other guy is in a different cage in the room. He's going to be listening to and responding to that song. And when this animal on the left is done, he will pick up and respond to his buddy. Good. So let's talk about the structure of that song. So you can see they were clearly singing for a long period of time. It's about 8 or 10 seconds per song. And that song, it has a specific structure. So it starts with these very simple downsweep notes and then gets longer and longer as the song carries on like this. So frequency of each note is shown here in kilohertz, time, and the duration of each note is shown with these dots. So think about on the right-hand side here, note duration. They get longer and longer and they kind of crest over like this and the animal stops. But what's cool is that they do this in an interactive context. They're singing back and forth. And so this is the singing activity of a resident mouse. So this mouse lives in a room. We've brought in a friend or enemy or another mouse. I don't know their relationship. They're both male. They don't have to be male. Females also sing. This other mouse seems to have singing behavior that's very well correlated to the white mouse. This is again over the span of an hour. Each tick here represents a bout of song. We can expand that out in time. So this series of 2 ticks is shown here to be the resident mouse in white singing and then the red mouse answering. Here's a case where the red mouse starts and then the white and then back to red. And we can plot this in the following way. The resident mouse will produce a song, and that's shown here. We look at the offset of that resident mouse's song, and when the other mouse starts to sing, okay? And we see that other mouse is engaged in this way. So this is a kind of very fast back and forth exchange called a counter song or counter singing. And this is found across a large number of species, but typically not species that are found in the laboratory. So that's something that I think is really special in terms of this animal and why to study it, because it engages in these very fast social exchanges. The one other animal that is reasonably widely studied that does interactive exchanges are these marmosets that do antiphonal calls. And those marmosets, however, have a qualitatively different time scale. So if we think about human speech, I showed you a histogram at the beginning of this, and that histogram was very fast, 200 millisecond response latency. The singing mouse is about 500 milliseconds, but that marmoset is about 5 seconds, okay? So it's a very different time scale and qualitatively does not match the kind of fast back and forth of human speech. What I think is true here is that the marmoset is leaning heavily on more kind of emotional circuits in the brain, things like cingulate cortex. If one stimulates cingulate cortex, and Asif Ghazanfar has done this, that causes activity within cingulate cortex, which then increases things like heart rate, and then seconds later causes the animal to respond with a phi call, which is shown here. So it seems to be a slightly different, though interactive, vocalization. So what's going on with this mouse and its cortex involved? So I'm going to show you a few causal experiments and we'll wrap up this talk. This is, again, the singing mouse. This is the cortex in the background of the singing mouse. And we stimulated around the surface of the cortex. And we found this area here we're defining to be the orofacial motor cortex. And there, with a very small amount of stimulation, we can cause very vigorous flexion of vocal muscles, things like jaw muscles or cricothyroid muscles that are actually involved in creating this song. We can then record from this hotspot of the brain, this orofacial motor cortex, And we see spiking activity. So this represents 28 simultaneously recorded neurons during the production of this vocalization. And we can take the activity of each one of these neurons and put it into this grayscale here. So this is 0 hertz in black and 20 hertz here in a light gray color. And you can see across these 28 neurons, from the beginning of song at green to the end of song at red, that things operate in a pretty fixed regime during singing. You see the same shape, the same patterns emerge across songs in this animal, but outside of songs, things are really mixed up. So it seems like the network, cortical network, is doing something important during this time. And also appears to be reflecting, to be modulated by social condition. So here's an example of a single neuron now. Instead of showing you 28 neurons, I'm showing you 29 trials from a single neuron. In this case, the animal is making songs of different lengths. Here's a shorter song in gray. Here's a longer song over here. And we can put that into a color plot here. I think it's a little easier to see, such that the beginning of the song is a black tick and the end of the song is a black tick. And there's some color here representing the firing rate of this neuron. And what you can see is there's some heterogeneity before the onset of singing. Okay, and if we plot this out on the context of this animal, if an animal's singing by himself, we see no activity before the onset of that song. But when the animal is singing in a counter-singing way, as a response to a partner, we see that partner's song represented in the firing rate of this cell. So there's a kind of social modulation that we think could be important in kind of getting the entire pattern going at the beginning. So this is an idea of a focal cooling manipulation. It's something that we've done to look at this, and I think the details are exciting, but I think in the interest of time, I'm going to leave this on the cutting room floor. I want to take you to a critical experiment. So our kind of model that comes out from this experiment is that there's orofacial motor cortex that could be exerting hierarchical control onto a subcortical series of note generators. And we think that this could be important for down here for generating notes. And this could be important for basically using those notes, deploying them in a socially kind of appropriate way. So that cortical input could stop and start, maybe change the speed. And we think that this could be really critical for countersinging, for interlocking those vocalizations with a partner. So we did the following final experiment. We had playback from a loudspeaker, and we could play songs of that same species to this red animal. And every time we play over the span of about an hour and a half, shown here, I'm going to show the timing of that playback with a vertical white line. And that vertical white line is often responded to with a red circle. That's when this animal responds. He's singing with the speaker back and forth. Great. Now, if we inactivate that hot spot, that orofacial motor cortex with mucimol, now the number of responses falls way down. But luckily, it doesn't go down to 0. And if we look at the timing of those responses, in red, under control conditions, if we just inject saline with no kind of psychoactive capabilities here, we see in those control conditions, the animal responds still quite quickly to a playback. But if we inactivate with mucimol, so we kind of temporarily make that area leaky, unable to fire action potentials, and now doesn't respond, we see that not only are there fewer responses, but they're quite late. So we think then of the kind of cingulate cortex, these more emotional circuits that are still online. We haven't turned those off. And maybe that's what's giving rise to these marmoset-like delayed vocalizations. But all the fast back and forth is gone. So that suggests then that, or I think demonstrates then, that this central orofacial motor cortex is important to then drive these vocalizations out in a social context. So the final piece here is we think about some kind of sensory trigger for interactive speech. some critical information if we're doing a task or other aspects of what a partner is saying that allows us to get ready and then respond to that partner. Here, we don't know what the specific features are of a singing mouse that trigger that response, but we're looking into it now. There's an interactive language hub in the person's brain that we think includes middle frontal gyrus and inferior frontal gyrus. In the mouse's brain, it's the orofacial motor cortex that we think is a really clear cortical bottleneck. And that leads to these fast-spoken exchanges in people and countersinging in rodents. And if we manipulate that, either by focal stimulation disruption here in people, or pharmacological inactivation in rodents, then we cause slowed interactions and linguistic errors, and we can abolish countersinging in the rodent. And so therefore, I think we can kind of learn from these animals something about that interactive speech and start to put together new models for how this works, and importantly, how to address this in cases where it breaks. Thank you for your time and your attention. There's a whole bunch of people to thank here, but bird team didn't really talk much about birds today. The mouse team, you got to see their faces, and the human team, which it couldn't have happened without the good folks at the University of Iowa. And thank you for your time and attention.
I hope you can hear our audience clapping, Mike.
I can. Thank you. Thank you all.
That's great. Thank you so much. That was both educational and inspiring, but also very enjoyable. We have questions in the chat room, and I'm quickly looking at our own room as well, if there's people who have questions. If not, I'm going to start in the chat room. And then in our own room here, where we're live, we may get some questions as well.
Great.
So the first one is a question from Aubrey Nunez. In how many fingers does a healthy person have? In how many fingers does a healthy person have? The informative answer is on the right of the structure, and this dislocation happens irrespective of the time dislocation. How does this fit with your model?
The informative, could you start with the word informative and read from there again, please?
Yeah, so the in that stimulus, in how many fingers does a healthy person have?
Sure.
The informative answer is on the right of the structure. So it's to the right of so it's on the right of the structure, and this dislocation happens irrespective of the time dislocation. I have to admit, I think Audrey is correct. So I think that in all of these cases, there's a lot of filler. And then that critical word comes in, and that's when the person has exactly the information they need to answer the question. What's true is that we don't just have 4 of these questions. We actually have 40 of those questions. So 20 different variants and 2 types. One where the word is listed early, and one where the word is listed later. So we could say a healthy human has how many fingers? And what's important is the planning dynamics follows that blue word around, if it's in the middle of the sentence or if it's at the end of the sentence. So we did that almost as an internal control for all of the other words in the sentence. So she's onto something, she's correct. It does start and then goes to the right. But we can kind of address any concerns related to that by moving the blue word around, and indeed planning follows the blue word. I hope that's clear. I would think in the, just to follow up, in the experimental setting, it does hinge on a certain similarity between questions, right? Where you're always asking about the normal situation or healthy situation, because otherwise one would have to think longer for the answer.
100%. And if I took you through all 40 of these questions, it'd be the world's most boring slide. But yes, indeed, it's the fact that there's a syntax to the question, a kind of overall structure that the patient gets used to and is listening for that critical word. Exactly right. Thank you for clarifying that.
Thanks so much. Next question is from Greg Hickok.
This line of studies is conceptually equivalent to the fMRI work we did localizing SPT, the spot, and the broader auditory motor dorsal stream speech circuit. Any thoughts on the connection?
I mean, Greg's work, of course, is extraordinarily inspiring to us and many others. The connection to auditory processing, is that the question? So I think there was a kind of concern from one of our reviewers about, does this have to do with comprehension? And so this is one way of thinking about the stimulation result. This is one way of thinking about the activity that we see. And so I'm going to lean on these experiments here, pardon me, as a kind of potentially useful control in that regard. So I think here, all of these questions need comprehension. They need the patient to listen to and understand the question. And yet, only some of these questions or some of these commands actually lead to activation within this part of the brain. So I think that it's in some sense a control for understanding or salience or comprehension, although he is absolutely an expert in this, and I'd love to hear his thoughts. And it'd be great if it could be passed on to follow up over email. I can learn a lot. I really appreciate the feedback.
Thank you. Had a question from myself. Even though the model and the data show a small extent of overlap between the perception, planning, and production stages in these lab tasks, they're still modeled as essentially sequential stages. And so what do you think is the particular process or ability that allows some speakers, well, all of us, but some speakers to a greater extent, to engage in all of these simultaneously, and in the extreme case, professional live translators, right, who listen and speak and plan all at the same time? So I hope my question.
I don't think it's firmly serial in a standard kind of ethologically relevant use. So if I'm talking and having to give an answer, as I'm doing now, that may last for 40 seconds, I'm not planning at the very beginning and then executing, right? These things are constantly online and constantly being used. So one, I think, very exciting future direction that we'd like to pursue is, and we're working with people who are into discourse analysis and in conversational speech to really look at that and try and dissect longer stretches of speech, right? And that aren't as kind of back and forth by design. And we do see, when we see those bumps, those kind of planning-ish bumps that happen, have you been to Mexico, New Mexico, this kind of thing. When the person is talking and there's an unfilled pause, we often see a big bump during that unfilled pause, right? So it's true that there's constant planning happening. For this is a very specific way of isolating that planning activity. But we also have done an analysis during conversation, and a lot of the tricks reemerge there too, right? So I don't think that things are firmly serial. They can't be, right? And that can't make sense. But I think this mechanism is there, and that we have to kind of understand step zero in order to understand how this can then be implemented in a more complex context. Does that make sense? Yeah,
it totally does. I think so. I think the reason behind my question is also, so that does pose a tremendous challenge for the system to segregate different signals that are coming at it almost simultaneously, right? You have to know what part of the sentence is this signal coming from, and then so 100%. And I can imagine that in stroke survivors, for example, that is the kind of fragile process that can be functionally impaired, right? Where people lose maybe that ability, or that ability is impaired to do all this at the same time.
So fully agree. I think that in looking at the cortical surface and looking at some of these functional maps, right, we do see spatial segregation. But even if the same area has activation in multiple phases of this task, it's a low resolution way of measuring the brain. We're used to looking at individual electrodes or as individual cells, right? And so how many cells actually give rise to a high gamma signal at a single electrode. It could be thousands, tens of thousands, hundreds of thousands. And so it's absolutely the case in simple animals that within a circuit, there could be distinct cell types that are mediating specific behaviors. And so it's absolutely the case, or it could be the case, that within an electrode, when one sees activity, that is a more nuanced set of cells doing things. And we'd love to be able to look at a cellular level, and we've taken steps towards that exact thing.
Thank you. There's a follow-up from Greg also, which I think both relates to his first question and also to my question just now.
Great.
Greg says there's plenty of evidence of sensory cortex involvement in speech planning. How does that evidence fit with the conclusion that perception, planning, and execution are separate? And that also, I'm speaking for him now. I think that also refers to the role of area SPT, for example, in integrating the two.
Right. So in fairness, we didn't get a ton of temporal coverage. We were looking in this area, and I think if we look in temporal lobe, and we can see, where he's clearly an expert, we can see what we see down there. I think I'm not ready to stick my neck out and say it's all distinct. I mean, I think that's a reckless statement, right? And could there be areas where things can be mixed selectivity? Of course, absolutely. And we do see certain electrodes that are mixed selective in our data set, right? But we also see some that appear to be distinct and actually do form some categorical clusters. So I think both could live together. I don't see there's a world where both ideas can't be correct.
Thank you very much. I had a final question on a different tack. So within the planning electrode sites that you distinguish, have you been able to distinguish between different functional levels of planning, such as semantic, phonological, and articulatory planning? So I think at some point you said, well, we don't expect articulatory errors, but there's also, at least theoretically, such a thing as articulatory planning itself.
Absolutely.
Right? Imagine.
Yeah, phonological, semantic. Yeah. There's a lot of different planning that's been described in the linguistics literature, and Greg Castellucci, my postdoc, is wanting to exactly go there. So he has now created a series of tests that is absolutely, they're designed to start to isolate different planning networks. So if one sees, for example, this area of the brain, one could think, well, those different types, semantic and phonetic planning, could be all intermixed, right? Salt and pepper. Or there could be distinct areas that could light up during this. And of course, we're specifically talking about planning within this interactive context. Planning is big, and planning can be done for a lot of different things. And I think it's something that fascinates Greg, and he really wants to go and do exactly this in the future in his own lab. So I think, and he's on the job market now, so hopefully that can happen.
Cool, nice plug. Thanks.
Yes, indeed.
Thank you. I'm looking at the room. Is there anybody here? Let's see. Then maybe I do have a final one. If you're willing to hang on for just another minute, Michael.
Absolutely.
In stroke survivors, so there may be problems with turn taking that are not necessarily sensory motor in nature, right? So your model is about the sensory motor aspect of this and of the distinction between perception planning and the motor response. But some stroke survivors may have problems with the psychology of turn taking. And I wonder if you have thoughts about how that interacts with this sensory motor aspect, whether you think there's a meaningful connection there, or whether those are just two very separate systems.
I think it's super fascinating. I think that in watching that movie, I've seen that movie a lot. I've seen a lot of these collections of people with expressive aphasia. He doesn't seem to have that much of a problem with turn taking. He's stuck with the words well, yeah, and no, right? So he's not able to access the correct word, but he can deploy something. And there's often, in the this literature obviously much better than I do, but in turn taking, there can be a laugh or a grunt or some other kind of vocalization that's given as a turn. that still, I think, signals some kind of a problem with access, for example. With respect to your question about the psychology of turn taking, also fascinating, and I'd love to maybe offline chat with you about, you know, where is the damage in those cases?
I don't know. I'm not an expert on that aspect at all, actually. But
okay, yeah. Well, I think it's super fascinating and interesting, and it's something that I'd like to learn more about. Yeah.
Thanks so much. I think that brings us to the end of our session, Michael. Thank you so much for your talk. I want to alert everybody to the talk on January 13th by Katarina Haley in the new year. And also, I'll be sending out an announcement for the call for papers for the Clinical Aphasiology Conference, which is held in May. So I'm using this as another plug. Thank you, Michael. Thank you, audience. And have a great day.
Thank you.